Children, today let's study about a very important process called fertilization. We are already aware of pollination. So once the pollen grain from the ripe anther is transferred to the stigma, that pollen grain germinates. When this pollen grain germinates, it forms a pollen tube. The tip of the pollen tube releases some enzymes which dissolve the solid tissue of the style and pave a way for this pollen tube to come down and enter into this region of the ovary, which is called the ovule. This pollen tube is bearing two male gametes which are discharged into this structure called the embryo sac of the ovule, where they take part in double fertilization. To understand what is double fertilization, we first need to understand the germination of pollen grain and the internal structure of the ovule. So let's begin with germination of pollen grain first. This is what an individual pollen grain looks like. Its outer wall is spiny and is called exine, and the inner wall is thin and is called intine. You can see that in between exine is not regular. It's not present in some areas in between. It's interrupted in spaces in between. And in these areas, it's been labeled, there are openings present here which are called germ pores. From the germ pores, the intine protrudes out, it comes out and it grows down and it forms a pollen tube. Now, what changes occur inside a pollen grain at the time of germination of this pollen? The pollen grain has two nuclei, tube nucleus and generative nucleus. The tube nucleus is the one which is responsible for the pollen tube, whereas the generative nucleus splits into two and forms two male gametes. So the two sperm nuclei or the two male gametes are actually formed by the splitting or division of generative nucleus, as the name says, to generate. And the pollen tube is formed by the tube nucleus. The pollen tube is nothing but the intine which comes out of the germ pore and grows long. Exine is the outer protective layer and intine protrudes out here and forms the pollen tube. Now this pollen tube grows longer and longer. The tube nucleus is also present in the beginning here, and very soon it degenerates and only two sperm nuclei or the two male gametes are left. It is these two male nuclei or gametes which come down here into this area which is called the embryo sac. To understand the internal structure of ovule, let's refer to this diagram. Now inside this, you can see inside the embryo sac there are 8 prominent cells. This middle cell is the egg cell or the female gamete. It is surrounded by two helping cells which are called synergids. Opposite to this egg apparatus, this whole thing is called egg apparatus. Opposite to this are present 3 cells which are called antipodal cells. And in the center there are 2 nuclei called polar nuclei. So egg cell, the synergids making up the egg apparatus, the 2 polar nuclei in the center and the 3 antipodals on the other side. Now what are they all doing and what is their role in fertilization? We know that male gametes are 2 in number and both are released inside this embryo sac. One of the male gametes fuses with this egg cell to form the zygote. We know fertilization is fusion of male gamete with the female gamete to form a zygote, and this is what is happening here. Whereas the second change which is happening inside this embryo sac is these 2 polar nuclei fuse together with one another and form a secondary nucleus or a definitive nucleus. They fuse to each other to form a secondary nucleus, and then they fuse with this second male gamete to form endosperm nucleus. So overall 2 fertilizations are occurring, one between the male gamete and the egg nucleus and second between the male gamete and the secondary nucleus. The product formed here is the zygote, which later changes to embryo, which will give rise to the baby plant. Whereas here, the product formed is the endosperm nucleus, which will later form the endosperm. And endosperm is that part of the seed which stores food for the developing seed. So embryo is the baby plant and endosperm is the nutritive tissue. So since in flowering plants fertilization is occurring at two places inside the embryo sac, we call it double fertilization. There are two polar nuclei fusing with each other and then fusing with the sperm nucleus or the male gamete to form endosperm nucleus is also known as triple fusion, because three things are fusing: the two polar nuclei to one another and then with the male gamete. So this is called triple fusion. Whereas the other fertilization is between the sperm nucleus or the male gamete and the egg together to form the zygote, which will change into an embryo. So in flowering plants or angiosperms, two sets of fertilizations occur, and that is why it is called double fertilization. And this is what has been explained here. This is a germinating pollen, a pollen tube carrying the two male gametes. Now this area of the ovule from where the pollen tube enters is called the micropyle. So micropyle forms a passage for the pollen tube to enter inside this area called the embryo sac. So you can check the labelings. This is the embryo sac, this is the micropyle, and the embryo sac is surrounded by a nutritive tissue called the nucellus. So nucellus provides nutrition. After fertilization, this entire ovary changes into a fruit and the ovule changes into a seed. These are the post-fertilization changes. So ovary to fruit and ovule to seed. Now around the ovule you can see protective layers are present here, two protective layers which are called integuments. These integuments only change into seed coat. We all know that seeds are covered by a seed coat having an outer hard testa and an inner softer or thinner tegumen. It is these integuments only which form the testa and the tegumen. So imagine this whole structure changing into a seed, the egg fused with the male gamete to form the embryo or the baby plant, this providing the nourishment by forming the endosperm, and the integuments forming the seed coat. The micropyle persists in a seed also. You will find that when we soak the seeds, they swell up, and when you squeeze a bean seed, you will find some bubbles coming out from an opening in the concave region. So the hole which is there responsible for entry of water or for exchange of gases is the micropyle. So micropyle remains, which was there in the ovule, it remains in the seed also. The integuments change into the seed coat, the egg cell changes into the embryo ultimately, the polar nuclei change into the endosperm. The antipodals have not been found to play any specific role in fertilization till date. The synergids are called helping cells. Sometimes it happens that the egg cell is not capable of fertilization. In such a case, the synergids or the helping cells take over, one of the synergids will take over the function of egg cell and take part in fertilization. So these are all the functions of the different parts of an ovule. You have to learn and even draw diagrams of different stages of germination of pollen, diagram of LS of ovule, and the diagram showing the process of fertilization, and also learn the fate of fertilization, which is mentioned here, that what all would change into which part. So you have to tell which part becomes what: ovary to fruit, ovary walled into fruit wall, which is called pericarp, ovule to seed. There is a placenta present here to the stalk of the seed, outer integument to testa, inner integument to tegumen, secondary nucleus which is formed by two polar nuclei into endosperm, the egg cell into embryo. If egg cell is not capable of fertilization, then synergids into embryo, and antipodals disintegrate or become disorganized. This is called the fate of fertilization. Different parts of the ovary, what happens to them. And overall we know ovule forms the seed and ovary the fruit. Here you are been explained what is double fertilization, which has already been explained to you. So double fertilization is important for examination point of view as well.